Alright, so we're coming into part 3 of this question. Part 3 of this video. Okay, for question 2, okay, we are looking at here, we have the European badger, which is a mammal found in England. They contract the bovine tuberculosis, which is caused by the pathogen Mycobacterium bovis, by the way. Tuberculosis as part of what we need to know under in our AS topic, which is topic 10 of infectious diseases. Researchers estimated that 5% of the badger population in England are infected with bovine tuberculosis, and BTB can be spread between badger populations and other populations of wild animals and farmed animals. Herds of dairy cows produce milk for human consumption. If a dairy herd becomes infected with BTB, the pathogen can be transmitted to human populations in milk. A variety of measures have been introduced in an attempt to reduce the BTB rate in badgers, so preventing the infection of dairy cows. And these measures include vaccination and also the removal of badger, which is fair culling. Alright, so let's look at the questions. Suggest how researchers estimated the percentage of the badger population that was infected with BTB. So I think there's two bits that we need to remember here. It's going to look at how we're going to estimate the percentage of badger population that was infected with BTB. Now, because we're looking at badgers, okay, I want to remember that these are mobile animals. Okay, they can move. Okay, so in our syllabus, okay, we know that we can use the mark, release, and recapture method of estimating the populations of mobile animals. So you can start off, okay, by describing first that the idea that we need to be capturing a large sample from that area. Okay, so large samples of badgers. And then following the mark, release, recapture, okay, you're going to be recording, okay, the number of badgers that have BTB, because we're going to look at the infected badgers with BTB, right? So after capturing a large sample of badgers in the area, make sure we record the number of individuals. Yes. Okay. And okay, we test those badgers. Okay, we catch those badgers, and then we test them with the we test the individuals to see if they have TB or not. Okay, we test the captured individuals of the population and record the number of infected individuals. And what we'll do next, and then we will divide that number of infected individuals by the population number and then multiply by 100 to convert it into percentage. Okay, there you go. All right. So what you can also use is that, I haven't mentioned the mark release recapture method. We can also use the mark release recapture method to estimate the population by using the Lincoln index as well. Okay. So basically describing how we're going to calculate the percentage there. So it is possible, so next bit, it is possible that wild badgers infect dairy cows with BTB. A group of scientists investigate the correlation between BTB infections in badgers and BTB infections in dairy cows. 30 study areas in England were identified. They are approximately 100 square kilometers. 10 out of the 30 study areas were randomly selected and given the codes of A to J. The number of infected cows and the number of infected badgers were counted in each area, which is in A to J, and then the number of infected badgers were determined by testing for the presence of Mycobacterium bovis in the tissue samples taken from these badgers, and then the number of infected cows were determined by testing for a response or inflammation to the introduction of antigenic materials into the skin. So basically those are the different ways of testing for TB. And then these are the data collected in table 2.1. We have datasets, two datasets only. We have datasets 1 and datasets 2. And between A to J, in datasets 1, we have A, B, C, E, F, G, and H, which were counted in the year 1999. And then we have D, I, and J, which were counted in the year 2001. The scientists expressed their data on the numbers of infected cows and infected badgers as the population density of infected cows and infected badgers, explain what is meant by the term population density, and suggest why the data was expressed in this way. So basically, we're calculating the number of infected badgers or the number of the population. Okay, so number of per unit area. Okay. All right. So why are we using population density? This is to allow for a valid comparison to be done. Okay. Between the two populations. Okay, because now they have the same units and we can easily compare the numbers. See, 3 variables that were not standardized in this investigation, meaning not standardized, what are not kept constant or not kept the same. All right, so first things first is let's look at the method, and we're going to be arguing what are the problems with the method that you should have kept the same. All right, so what they didn't mention us is that number 1, okay, we can notice that, okay, yes, they did mention that they studied 30 areas. That's fine. All right, they mentioned the area which is that's being covered, which is around 100 square kilometers. They did some random sampling, okay, but what we can argue here, and let's look at the data, okay, in this area, we notice that, okay, the year of which this population were counted are not the same. So that could be that could be a problem too, because what we actually do want is we should be standardizing, okay, when these measurements are being taken. Okay, so the year counted were not the same. Okay, that's the first argument that we can make. Okay, that should have been standardized. But they didn't. Okay. They also, when we're looking at the testings here, they didn't mention about what cows or what species of cow, what breeds of cows are being sort of included in the investigation. So that could be a problem too, because there are different species of cows as well. So we should keep the same breed of cows to be tested. Okay. The testing method was also another problem here, because they mentioned that for the badgers they use tissue sample, but for the cows they use different testing methods. So that could be another procedure that could have been kept constant. Okay. Method of testing for TB as well. Okay. Alright. And then when we look here with the two data sets, not only that the data were counted or the population were counted at different years, we don't have the same numbers of data sets. In data set 1, we have 1, 2, 3, 4, 5, 6, 7 data sets or 7 areas counted, whereas the other 3, which is D, I, and J, were counted in a different area, so at a different time as well. So that could be a problem. So the number of counted areas as well, that could or should have been the same or kept the same or standardized. Okay. All right, so those are the possible points that we could put here. Okay. The scientists decided to analyze their data on the occurrence of BTB in cows and badgers, statistically state the null hypothesis. Now we were looking at correlation, so I think that one is quite straightforward. Is the idea that there is no correlation between the number of infected badgers and the number of infected cows. Okay, just like any other null hypothesis, since we're looking at correlation or relationship, any null would be there's no correlation or no relationship as well. Okay. The scientists plotted a scatter graph. The scientists plotted a scatter graph of the population density of infected badgers and infected cows. The scientists drew a line of best fit, interesting, on the scatter graph. And then we have here on the x-axis the number of infected badgers per square kilometers with the number of infected cows per square kilometers. Okay, fine. We can see a line of best fit here, somewhat showing an increasing trend. That means there is a positive correlation, just quickly looking at the best fit. Okay, so the scientists carried out statistical tests on the two data sets combined and on each set separately, and the results of these tests are shown in table 2.2. all right, so from here we have table 2.2. we have data sets where it's combined and separate. Okay, interestingly, we can already look at the pattern in this graph, that when we look at the two data sets combined together, it does show a positive correlation, and it seems to be significant at probabilities of 0.05. Okay, that's fine. However, when we look at the data sets separately, here when we look at data set 1 and even data set 2, they are actually showing a negative correlation. However, okay, even though it does show a negative correlation, this is not very significant. This negative correlation is not very significant at probabilities 0.05. Okay, that's what we can sort of really quickly tell just simply by looking at the table itself. Some scientists suggested that this information showed a strong positive relationship between the number of infected badgers in an area and the number of infected cows in the same area. These scientists also suggested that the BTB in herds of cows could be reduced by removing badgers through culling. Okay, evaluate whether or not the data from figure 2.2 and the table 2.2 supports the suggestion that the scientist has made. Now, when we are asked to evaluate something, that means we need to find out points that do support the data and where exactly does that data is showing support and where it does not support as well. All right. Now, we want to find out, let's just try and find the first bit, which is one that shows a strong positive relationship. So let's find out the data that does support. Okay. Let's find out where exactly. Strong positive relationship, that means we have a strong correlation. So from the table and from the figure, okay, we can see that there is an increasing pattern here from the line of best fit. Okay, even from the data table here, we can see that for both data sets, there is a strong positive correlation, and that is quite significant at probability 0.05. So the combined data set seems to show a strong positive correlation. So that is one way of how support the data does support. Okay, and then we can also see that here from A. So for all of these data, okay, in fact here, okay, that we can see that increasing. Okay, so as the value here, we can see that as the number of infected badgers increases, okay, we can see that the number of infected cows also increases for the data sets. Okay, so which means that we can actually use this information to support the suggested information, what the scientists have suggested here. So it does show a strong positive correlation. All right, so we can use that. Yes. Sorry. Okay, and also the table. At probability 0.05. Then we can argue for the other bits why it's not supporting. However, suggestions include. Okay, so the one that does not support is the idea that yes, even though when we have the combined data set, it has a positive correlation, and moreover it is significant, when we look at the data set separately, they are actually showing negative correlation. That's the first one. The second one is the idea that, however, we know that if you look at the presence of these data sets, okay, D, I, and J, so this is data set 2 and this is data set 1. Even though the graph of line of best fit is quite nicely done there, but we notice that there is a clustered of data. Okay, so this is what happens in for data set 1 in the graph, and then this is what the pattern that we're showing for data sets 2. They're actually quite clustered. All right, so it's not nicely distributed. All right, the graph. Okay, so we also need to remember that the data is probably due to the fact that these readings for different data sets are taken from different years. So that may not be supporting the correlation, obviously, because it is showing a negative correlation. Okay, so you can add that as well. Did not support the suggestion include. Okay, number 1. So they're clustered from the graph, but also negative instead of positive correlation. Although it's not significant. Alright, so explain whether or not the data support the suggestion design days match. So we've mentioned the bits where it does support, and we've also mentioned the bits that does not support as well. Alright. Okay, so that's the end of the question. I hope that is helpful, and if you have any other comments, do leave your comments below. And I'm sure, like, if you notice that if you have any suggestions that you would like to share with me, or if you have any requests, just feel free to send me or leave a comment, or just send me a DM. And hopefully this is helpful. Thank you very much, and good luck for your revision.